which is used for the digital holographic microscopy and interferometry. And then I started the working on the same digital holographic microscopy for biomedical application, and somehow we find that the interesting area, why don't we go for single cell analysis. Okay, instead of when we have the smear microscopic slide and smear where we have the different layer ups blood cells present, and then we generally visualize under the microscope, right? And we see that how they are the cells, and depends upon that we will be finding out how many white blood cells are present, how many within the field of view, how many red blood cells are present. Based on that, in clinical microscopy, we used to do the testing, right? Diagnosis. So instead of that, we started in the working on the microfluidic platform. So others they are working with they are also working on the imaging aspect, and so I have started slightly divide and debating with them why don't we the find both the information uh, like uh, single cells, their morphology, as well as the change of the intensity of each and every cells. So for that, we have I have utilized this microfluidic platform. So design and development of the single microfluidic device, a single cell flow microfluidic device, and then the realization of the point of care device. So as we know, the most of the the diagnostic instruments are you can see in the diagnostic centers, and they have how generally they works, right? So mostly we have the different type of the instruments is available in the same place, and then like if you are the sick with the fever or something, you are having the some fever or something different any disease the sickness. So we used to go there and give the blood sample, right? So but the point of care diagnosis and point of care device development approaches came why should the patient supposed to reach to the the diagnostic center? Can we take this instrument to the actually who need, right? Instead of we have to go to the the centralized facility where we have the diagnostic center. So based on that, I have started working on this area, and my thesis title also more or less the same, absorption flow cytometry for point of care diagnosis. So basically, flow cytometer means it is indicating single one instrument, right? So single one instrument. But we have introduced that the single cell based analysis, which is mostly work on the absorption based. So that's the reason we called it is the field. Okay, so that we can use different biological samples which are uh, their response with they respond uh, based on the change of their absorption. So like that various different applications we can use. So that is the idea behind that the field we have introduced. So with that, with this I will start here. So as I started my topic is absorption flow cytometry for point of care diagnostics. So as we are aware about the each and every term here which is you are using. So the absorption is we know, right? Suppose if you have light, uh, uh, you have light and the sample, right? And here you have the detector. So detector, then whatever suppose whatever the light it is coming from here, and the sum of the light we are losing here, and the remaining light is reaching to the detector, and which is we are finding through the electronic components and other things, which is the input is photons, and output is electrical signals. Either it is a current or voltage, whatever we are taking with respect to that, right? So the amount of the loss we are losing here, that may be due to the sample absorption, right? Or the various other like the other ambient this surrounding due to that like the light loss. Maybe it is scatter information also. But so when we have like this and which is like when you have the bulk sample and you are doing such measurement, then whatever the information you are getting is like the, it is an average information, right? So instead of that like why don't we like the find the single cell and its absorption properties. That was the idea, and like the how it was realized, this technique I would be presenting here. So yeah, so before going to the talk, like I would like to stress here something small. So absorption flow cytometer for point of care diagnostics, but why we need to go for this? What are the advantages we have with the point of care devices? So here we can see a couple of devices. For example, this is like the path shoot. This is like one instrument. It can give almost 16 different tests. Okay, so 16 different tests, and like that only cytochroma and the self-testing diabetic monitoring devices. So I don't have any relation with this, like, but these are the some of the compact devices which can be taken into like the where actual need it is required, right? So if you see some of the advantages, we can test at point of care, and these instruments are portable, and more or less whenever we are having these portable devices, it works automated, like the when you give the some sample, right? It suppose you pick the blood sample and put here, and it detects automatically like the when you on the switch on the and off, right? So with that, like the, whatever we have the internal algorithm which is developed, based on that it works. So it is automated, and when your device is compact, when your device is small, okay, the cost also automatically go down, right? So and obviously the point of view devices are user friendly, right? User friendly means like when we have like the, using the some buttons to on and off, and you are like able to see that are the your results is displayed. So then automatically they are the user friendly, and the response also you will be getting the quick. So this is the reason like the most of the works like even though today's like the previous session uh, today's previous session was like okay, so today's previous session also mostly talked about like the POC, right? POC to POCD, right? So proof of concept to like the proof of concept to the point of care device. So this is the these all are the reason like the why the people looking towards like the developing of point of care devices. So development of the absorption flow cytometry for point of care diagnosis or diagnostics. So how we have got the initial thought. Okay, if you see the basic principle behind this, actually, it is similar to the flow cytometry, and we are very much experienced about like how the flow cytometry works, right? So if you see here, generally these are the cells which are flowing, and surrounding is the sheath is present, right? So when cells passes through the laser beam, okay, when cells is passes through the laser beam, so we can see here like there is a like the light is scattering, okay, light is scattering, and like the direct light is reaching to the like the here one obscuration bar is there, that is this is a blockage, okay. So in surrounding, if you see, this is the detector. Okay, if I say it PMT, actually, photo multiplier tube. Okay, this is also another PMT, photo multiplier tube. Okay, PMT. Okay, so so what you are seeing here is like the light is scattered when the cell is passing through the laser beam, and that scattered information is detected with PMTs. Okay, so whatever you're seeing here, this is the forward scattered information, and these are the side scattered information. Side scattered information gives you I think like the morphology of the image, and this one will give the change of the intensity. Like the one side is gives the size of the cell, and another is like gives the internal structure and other things. Okay, so this is general like the any flow cytometer working principle. Okay, so of course we will be having lot of in background like the lot of like the components, like which is the optical components and the optomechanics and electronics components also. So to get the electrical signals at final we are going to get the electrical signals only, right? So here these are the photons light intensity, and this is the laser beam. So this is a general scenario how it works, and the flow cytometer works based on the scattered information. Okay, so uh, we thought like the so basically here like the you would be using the high power laser source, and if you see each and every PMT they itself cost around around close to 2 lakhs. Okay, each PMT. And when we are having the like the flow cytometer, which is like the has the multiple PMTs and the laser beam also the high cost, and each and every component is very costly, like they are very expensive. Okay, so why don't we like the use a simple technique, that is the absorption property, instead of the like the scattered information, so that we can modify our design. Instead of the high power laser, a simple like the diode pointer, laser pointer we can use. And instead of the PMT, which is like they are having the very high sensitive, instead of that, like the avalanche photo detector, APD device, detectors, why don't we use? That was the initial thought to like the work towards the realization of this absorption flow cytometry for point of care device. So for that actually, and it was realized in the like the after completion of the work, and so it was realized as the absorption flow cytometry. And that too, instead of the complex like the structure what we have here, like suppose if I show here, so you can see this is the like the integration of like the optomechanics, optics, and laser and electronic components, and various detectors, everything will be there. Okay, so that was the third. And if you see like the flow cytometry, when you are like having uh, the flow cytometry but fluorescence flow cytometry. Okay, so in that case you are getting here the straight light is the normal, and here depends upon the like the excitation and emission of the cells. Okay, so suppose we are a laser source is the excitation, laser light is exciting this cell, and these cells will be emitting, okay, emitting the different wavelength of the light. Okay, with respect to of the each and every like the wavelength of the light. Okay, you can see here these are the different filters, which is also called dichroic mirror. Okay, dichroic mirror. So you can see here this like the dichroic mirrors will do like suppose uh, they would be having some bandwidth. Okay, for example, I will talk like this. Suppose you have uh, one dichroic mirror which has like the 520 plus or minus 10 nanometer. Okay, which is a reflection, and remaining light is transmittance. So it means like the reflection and transmission. So whatever the light is coming to reaching to this point, okay, 520 means like the 530 or 510 nanometer of the wavelength of the light will be reflected, and the remaining any wavelength of the light is like the reaching to this direct mirror, it will pass out. Okay, it will pass out like that, you can see here. Okay, so this is the like the actually fluorescence flow cytometry. Okay, so based on that, the analysis will be carried out. Okay, so yeah, as I initially said that instead of this complex structure and so on things, why don't we use the simple technique, that is the taking into the absorption properties as a like the detection parameter. Okay, so so that no need of any reagent for the test, and just sample you can flow through it with the help of the like the microfluidic platform. Okay, so with the help of the microfluidic platform, and you can get the signal related to that. So that was the way. And so this talk mostly like the covers how like this absorption flow cytometry or absorption flow cytometer, okay, instrument is realized. What are the steps involved here to characterization part and the experimental design and other things. Those all details I'll be talking here, and like the various what are the applications we have implemented and what are the other applications it has. That information also I'll try to cover here. So outline of the presentations, if you see broadly, like the initially I will be discussing about like the different type of the instruments, how they works, and what are the different type of the instruments are available, and like the where my instrument stands, where we developed instrument will be standing there. Okay. Then I'll be talking about my first work, which is the single cell absorption measurement and a blood smears. So basically here, why we have started with this, like the, this is more or less, it is called like the proof of concept. Okay. So proof of concept in aspect of like the, like how generally we used to see like the spectrum of like the absorption spectrum of any like sample, and if you do like the single cell absorption, okay, how it will be changes. For example, you take so one drop of blood sample, it is consist of like the several like the few microliter of the blood sample, right? And if you are like the how many of you know or not, I'm not sure, but most of you are aware about that, that a 1 microliter of the blood sample contain, okay, 1 microliter of the blood sample contain more than around 5.5 million red blood cells, right? 5.5 million red blood cells, and suppose around like the 8 to 12 thousand, okay, 8 to 12,000 white blood cells, WBCs, and close to 2 to 4 lakh, 2 to 4 lakh like the platelets, right? So platelets. So so if you are having one drop of blood sample, okay, it means it is having like a huge like the amount of the like the billions of the cells, right? Billions of the cells. Uh, uh, Actually, I'm wondering like my presentations, my slides are visible.
Hmm?
Hello. Yeah,
Yes,
am I
sir.
audible?
Yes, sir.
Yes, sir.
Okay. Uh, some participants they are actually unmuted their self. Okay, fine. I think now it is okay. Okay. So anyhow, so yeah. So okay. So you are having the suppose like the absorption spectrum, how you generally measure, like when you have the cuvette and the blood sample will be there, and the light source is generally illuminating it, and you have the detector at the second end. Okay, right. So whatever you are getting here is the average information of this million or billion cells, right? Instead of that, if you are able to measure a single cell and its absorption, so whatever the your instrument you are developing, okay, that would be really like the high sensitive, okay, and accurate information you will get. So initial my past work is involved like the design and development of the experimental setup for measuring a single cell and its absorption. Okay, how we realize like we developed a experimental setup, and then like how we measure, okay, single cell and its absorption. So that will be discussed and then followed to the like so here like the we are able to measure the single cell and its absorption by using the whatever the custom developed system, okay, but it has some limitation, that is the throughput. For example, when you are having a smear, uh, means you need to suppose smear is on top of the microfluidic microscopic uh, what you call it, microscopic slide, right? Microscopic slide, and you need to prepare this slide, right? and then you have to visualize under the microscope. So similarly, when we are able to measure the single cell here, okay, so but it is the throughput is the issue. So to overcome that, we utilized this microfluidic platform so that from smear to inflow with the help of the microfluidic platform, and we are able to measure the single cell and its absorption property, okay, at high throughput. And the throughput was it was close to almost like the yeah it was close to something around 3000 cells per second or something. Okay. uh, no, yeah, something around like in 1 minute we are able to measure almost 2 lakh cells per second, 2 lakh cells. Okay, it means 200,000 cells. Okay, so to realization of the high throughput, the single cell analysis, microfluidic platform will is developed, and we are able to measure that. Okay, so this is the second work which is I'm going to show here, and then I'll be talking about the actual thought, that is the source sample detector, the realization of the absorption flow analyzer for point of care diagnostic. Okay, so I'll talk about this like a simple small laser pointer is used, and the microfluidic device used for the single cell flow, and the APD avalanche photo detector is used for signal data collection. Okay, signal collection of the like the cells when they are flowing through it. So then I'll be talking about like the miniaturized prototype for the clinical evaluation. So actually, I have stopped work here, and so with this my PhD was done, and I came I went back to the some of the job, and there the work was different, so I couldn't pursue that again there, and so like when I came back here as a faculty, okay, so I'm pursuing this again. Okay, so that the technology is aware, and so now I have to replicate it and then look for the clinical evaluation. So this like the how it was developed, I will be talking here. Okay. Then the some of the other applications what we can add into this, like the optofluidic hyperimaging system, optofluidic hyperimaging system, as well as like the POC, absorption flow cytometer for point of care device. Those I will be talking about that, and then some of the other applications related to this work I'll be talking. Okay. So coming to the first thing, as we know, uh, any healthcare system which is used for detection, and that it has to it has to be developed it has it involves like the development of various diagnostic tools and technique, right? So of course, if you want to like the screen and the eradicate or the eliminate any diseases, it is not easy. So suppose you have the one system or one technique, okay, you cannot utilize it for everyone, right? So there is a need of like the various techniques supposed to be developed, and different instruments supposed to come, so that only like the when it is available for each and every individual, like each and every places, okay, so then only it will be you can get it exactly like the actual need of that actual purpose of that instrument will be served. Okay, so anyhow, this works is based I'm going to present here is the development of an instrument platform, okay, which can work based on the optical properties. Okay, it might be the absorption or transmittance. So of any biological cells, so which is like the react with like light. Okay, so for example, absorption, there are like the more or less both are the same thing. Suppose if you are measuring, suppose my light is here 10 milliwatt, okay, and at that detector I am measuring here, okay, 9 milliwatt. Okay, I'm talking in general perspective. Okay, or like you can consider in the microwatts and so on. So this detector is able to, means here whatever we are having the loss. Okay, due to that, maybe due to the path length and other sample absorption and other things. So if you are measuring this, then it is a, like the transmitted light, whatever the transmittance happen, that light you are getting. But if you are measuring that, like the whatever the lost light here, that is the like the 1 milliwatt, right? 1 milliwatt, which is supposing in example like the 10 milliwatts is your source at the source point, and the 9 milliwatt at the detector point. Then what if you are measuring this, then it is the absorbed information of the light, right? Absorbs information of the light. But if it is a transmittance, then like, if you are measuring this, then it is a transmittance light. So development of this instrument is a this developed instrument is a portable and field setting system which can work in remote places where the resources are limitedly available. So basically, as we know, when we talk about the remote places or the agencies or rural areas, okay, so we don't get the any sophisticated like the diagnostic centers, right? So and we don't also get like the highly skilled technicians. So like any like the peoples are available, but they are not so highly skilled. Okay, for example, if you are looking for the different uh, uh, the layer of like the smear, okay, they cannot do such kind of the if you are they are not expertise. Okay, so these portable devices, like the whatever we are going to talk about that, this will be work in the remote places and limited with limited resources. Okay, so to develop this instrument, okay, for the characterization and validation of the like the technique initially and the instrument system which is we are developed. Okay, we have chosen a specific like the disease that is malaria initial development purpose. And as we know, like the malaria is one of the deadliest disease in the developing countries, especially. And if you are like the how much you are aware about the uh, what is that World Malaria Report? Okay, suppose if you see 2018 and 2019, sorry, 2019 and 2021-22. Okay, it was like till here it was like the it is going down, but due to maybe different like the uh, effect like the COVID pandemic and other things, again these malaria cases and estimated cases and deaths are increased. If you see 2020 report, 22 report, okay, it is like close to 258 million cases were estimated, and almost close to 6.5 or 7 lakh deaths. Okay, so earlier it was came down to like the 5 lakh actually. Okay, throughout the world I'm talking. Okay, so if you see specific to our like the our region in India, okay, or the Asia region. So from India only we are like the seeing close to 82% cases, and the estimated cases and the deaths, everything is like the coming from the India only. Okay, so here there is a need of like, even though we know we have some of the like the point of care devices like RTT, rapid diagnosis test and other things. Okay, we need to develop many more like the devices which can reach to the remote locations and other places. Okay, so coming to the like the worldwide, if you see still around 87 countries are affected with the malaria. Okay, so if you see malaria diagnosis, okay, which is basically involves identification of the malaria parasite or its antigens related products on the blood of any patient who is having the patients. Okay, although when you see it, hello, someone, okay. So someone is like the unmuting themselves. Kindly don't unmute you. Sorry, it is getting disturbance. Okay, fine. So if you see diagnosis of malaria, identification is basically the parasite detection or its antigen related information you are going to get. So when you see like the in the like the overall, it looks simple. Okay, you have to see one like the parasite which is present in the blood sample, right? So, but it relays in the various things. So, like the it will be depends upon the different malaria species. There are different type of the malaria species and the stages of the infection, and many more things are there. Okay, so like the endemicity and like the interrelation between the like the level of transmission, immunity, the person's how much he has, and suppose the transmission. Suppose if one person's having the malaria, okay, and he is like the one mosquito bited him, and that same mosquito is like the fly to the various places, and that mosquito is bited to the different persons, then all of them will be affected with the same like the malaria, right? So so that is comes under the transmission. So as we know today's scenario, like the how the mosquitoes are everywhere. Okay, if you see in a specific the places like if you go to the villages and other places, so generally you will find in the specific a particular season you will see that these mosquitoes. But coming to the cities and other places, throughout the year you will see. So in my personal view, whenever see the mosquito, okay, so immediately my mind goes to the okay, which is it, the male mosquito or the female mosquito? Okay, and like maybe it is with malaria infected or normal one. So like that it used to strike into the mind. Okay, so like this all affects. So if you see different diagnostic approaches up to malaria, malaria or any other things, as we know, clinical microscopy is like the gold standard approach. Okay, so in the process of the clinical microscopy, if you go through, you can see the pricking. Initially, you can clean the fingertip and prick with needle, right? Prick with needle. Then you will get some blood outside, and that can be taken onto the microscopic slide. When you get it in the microscopic slide, then you prepare a smear. Okay, depends upon the like your requirement, thin smear or thick smear. Then once the smear is prepared, then you have to stain with like the methylene blue or the are there any stains? Like for example, you can see here the gypsum stain. Okay, so and then you have to make it to the dry it up. Then there is a subsequent washing steps. Once you wash that, then like the whatever the debris and other like the unwanted stain which is you have given to the like the, on a microscopic slide, that will be washed out, and then you have to see under the microscope, right? So then you can visualize like this. This is a field of view, okay, particular area where we can see, and like the if suppose this is a parasite which is present inside the RBC, okay, and this is the like kind of the debris or like the different things, and this is the parasites. Okay, so depends upon the like the suppose you can see here this is only small one, but coming to this like the RBC, you can see like the multiple parasites or the multiple stains. Okay, here you can see the ring stains. So this is like various like it shows you like the stages of the infection, like the what is the severity of the malaria, and the what is the percentage of the blood cells are affected with. So you will be seeing with this, and still this is the gold standard method. So basically this process generally when you go a thorough like the approach if you take, so it takes generally 1 to 2 hours. Okay, and if the some people can do quickly, so but there is a misdiagnosis possibility, so it will take the lot of time to do the single test. Okay, so a few more things are there related to that, the diagnosis of malaria. Okay, so yeah, so even though I don't have here that slide, so to alternate this, okay, to alternate this, we got like the RTD, right? Rapid diagnostic test. So this is a strip-based test. Basically, you will be having one strip, right? There, like the one is control and one is test. So control is generally when your sample is flowing through it. It is like the kind of the related to the antigen, like the suppose whatever the you pass. So this is already pre-coated one. So both the things. Suppose like you are getting here only one strike is the color change takes place, but here it is not checking. Then it is the negative test, right? So the person is not having the malaria, but if this is also get the color change takes place, okay, then it is a positive indication. So when you get the positive indication, so what is the different like the which type of the parasite is present and what is the like the different species is present and other things. For that, you have to go back to this clinical microscopy also. So then only you will be like the getting the proper information. So that is the status of this. So as of now, actually, this is highly used for like the you can get the quick test report and other things. But the efficiency of this system is around 70%. Okay, if you consider this clinical microscopy is 100%, this is the 70%. Okay, so some of the like the other things which is the instruments which can be used for the diagnosis of malaria. That is also like the it works related to like the based on the its antigens and the products related works like the PCR polymer change reactions where like the suppose you have something one molecule or one thing so like it will be multiplying that into the multiple different cycles and you will be getting that related information. So coming to the PCR analysis for the malaria diagnosis, generally it is a kind of the contamination process. Here, like the very high sensitive, even a small contamination also misdiagnose the like the report and the other things. Okay, so that is the problem here basically, and it is also time-consuming process for the in the specific application oriented. So some more instruments are there like so one is the flow cytometry. Okay, so one is the flow cytometry and another is like you can call it like so yeah flow cytometry and one more is the okay this is the okay so flow cytometry. So initially we have talked so flow cytometry or the flow cytometer the instrument and flow cytometry is generally called as a field. Okay, so uh, the flow cytometers can be used for various applications, including the biological cells, like the blood cells and other things for us, and the plant cells and many more particle analysis or very more. So it depends upon the suppose if you are adding some fluorescence dye or related to that, then it will work for the fluorescence like the flow cytometry. Otherwise, is the normal like the scattered information flow cytometers generally. Okay, so we can get here, and if you see, this is generally used like the cost of this instrument is like the 50 lakhs to okay 5 crores something. Okay, so generally different depends upon like the how many photo multiplier tubes. Okay, how many photo multiplier tubes are you are using? What are the different type of the source you are using, light sources, and what are the other like the components you are using in that? So the cost is high, and the bulk instrument it cannot be taken into the like the as a point of care source, and so it is a laboratory clinical diagnosis approach. Okay, we can use this for the analysis. You can find the we can use, but the malaria test for purpose it is not using. Suppose if you are using RDT, rapid diagnostic test, which is costing around 50 rupees. Okay, you have to pay here for this flow cytometer test. You have to pay like the 5,000 or the 500 rupees. So there is a huge difference in that. So that is the reason it is not used, and like the highly skilled persons also required to add the reagents quantity and the sample preparation and other things. Okay, so Another advancement was happened here. Whatever you get is a change of the intensity quantity, level of the intensity quantity. You can suppose you are getting here, this is the detector, if you are getting here the like the 10 microwatts of the like the light, it is like the intensity 1D signal, right? 1D signal. So to overcome that, if you want to see the morphological features of those cells, these cells, suppose whatever you have here, the white blood cell, okay, and it has some internal structure, you want to see these, you can get some change of intensity, different level of the intensity based on the size and other things, but you cannot see the what is the morphological features it has. To address that, actually imaging flow cytometer came into the picture, and this can serve like the in a larger manner and the high speed, high throughput analysis, and so on, and the cost of this instrument. And so particularly we can say it is, this is the imaging and image based analysis approach. It is okay, image based analysis approach. So you can get the test, but here also if you see, it is also close to 5C. Okay, the cost of the instrument and the bulky, and the cost per test also high. Okay, and you need the different reagents to identify those morphological features. Okay, so this is the reason actually it is not in use for a specific malaria diagnosis and other things. Okay, so if you want to see this different like the few more like the different tests. Okay, so we have mentioned here the RDT test. This is nothing but the rapid diagnosis just for malaria. Okay, and the QBC, quantitative Buffy code test. So here also you can see identify, but it needs to visualize this like the morphological features. You need 100x like the micro objective. Okay, so so that you can find out that like the red blood cells, and if you are they are infected and other things, and so generally it is a plasma range and the platelets layer. When you do the some centrifuging this like the cuvette, okay, you can find the different layer of blood samples. Okay, so this is like kind of the plasma. This is not like we are not in considering, and the red blood cells which are like the settled down in the concentration. So here this is the area of interest where we can find like the malaria infected cells and other any infected cells. Okay, so this is the one area like the, so depends upon the stages of the infection. If it is the highly infected, then you can see some the layer upper layer, but it is like the very early stage, then you can see here, because we see a small pigment of like the malaria parasite here. Okay, coming to this RDT, rapid diagnostics, more commonly which is we are using as of now, you can see here the sample you have inserted, and maybe it is the buffer play, like the sample you have inserted, and you can add buffer here, and that sample will be going through it. So for example, this is the control and this is the test. So this is the test like the bar. Okay, both are getting like the taking place of the color change, then it is the like the malaria positive, right? If it is only one is coming into the picture, then it is the malaria negative simply. Okay, so of course how many of you, and this is the outlet, right? Outlet means like the when your sample is entering in, like you are inserting from here, and like that sample will be coming out or the storage here. So anyhow, so this is the one, and this if you want to see about this, and if you open this uh, the case, okay, you will get, you will find a simple strip that is called actually paper based microfluidic actually. So here whatever you are using, that is the paper based microfluidic. So basically here the test bar and the control bar, you will be having two like the antigen or the antibody which is coated, and when the sample flows through that, whatever we have the antigens which is passing through it, then that it takes place of the color change. Okay, so as few of the point of the devices was evolved in a past decade, past one decade, maybe you can call it, or like the 1.5, almost 2 decades, we can say. So here we can see cell phone based malaria detection device. So basically you would be preparing some malaria microscopic slide and you are visualizing with the help of the cell phone camera. Okay. So you can find that like the different morphological features about that. Again, you need to prepare here the like the microscopic slide, right? You need to prepare the microscopic slide and you have to visualize and you need to develop a algorithm which can identify the morphological features. So again, you can find here the like the limitation is like throughput is the again challenging. Okay, so to address the throughput, okay, so you can see like the instead of the smear based analysis, you can find here like these are the like the some of the like the sample flow through the microchannels and like you can find here the malaria parasite which is present in the RBCs. Okay, red blood cells. Okay, this was also developed, but if you see the limitation of this instrument, of course, here the advantage is the microfluidic device you are using for the like the sample flow, right? And just like the methylene blue or the like the Giemsa stain you are using for the staining those blood samples, and you are able to flow those blood samples, and you can identify the things whatever the changes takes place. Okay, so the limitation here is suppose any mobile camera, if you see, okay, it has 15 to 30 frames per second. Okay, 30 frames per second. This is the limitation here actually, generally. Okay, so to address that, okay, to address this I think I'm running behind. Okay, I'm late. Okay, so I'll quick now. Okay, so to address this, actually here also this 30 frames per second is the maximum, so you can not get like the more information here. So if you see the overall flow chart of the different instruments which can be used for the malaria diagnosis, we can broadly classify into two categories. One is the rapid, that is the fast. You can 5 to 10 minutes minutes can give you test information, and the one more is the quantitative here. So what it is, this rapid test will give positive or negative information only, it will be providing. Okay, coming to the quantitative, you will be getting that informations like suppose how many cells, okay, cells are infected out of the total whatever we have the with respect to the region of interest. Okay, so if you see further like the in the quantitative, okay, so here also you can see RTD and the BPC course, these are the two categories. And similarly if you see here, so microscopy based analysis, or you can say that morphological information you will be getting here, right? Morphological features of the like the cell and cell information you will get here. And one more the category we can classify here is a detection based or the non-imaging system. Okay, non-imaging means like here you will be getting the change of the intensity only. So further you can see in the microscopy like the bright field microscopy, fluorescence microscopy, and many more like confocal microscopy, two photon microscopy, and many more we can have. Like it depends upon the complexity we can find. So further you can classify it into the like automated or conventional. And if you go automated as well as the bulky as well as the point of care, conventional means it is a conventional microscope which is we are using. Similarly here also, so based on the detection analysis you can find here, based on the change like the identifying the DNA RNA like the informations you can find, that is the PCR and other thing, right? PCR and other thing, and some of the optical properties. Based on the optical properties changes, we can find out like the some of the different techniques. Okay, so there we can see like the other optical methods we can use, like the spectroscopy, Raman spectroscopy, and many more other techniques are available. Okay, so my work also lie down here only in this area only. That is called absorption flow cytometry. Okay, absorption flow cytometry. So if you see more or less, I'll tell you one thing here. Either when you are like the sample preparation procedure is each and every instrument is required. Some of the instruments might be needed like the pre-sample process. Suppose you prepared some device. So when you are preparing device, during that time itself you have to do the some sample preparation so that you can directly use the samples to test it. And some of the techniques is you need and the post pro like the in the pro like the when you are conducting the test, then already you need to add some of the fluorescence dye or the methylene blue or other anything. Okay, so more or less every instrument is required. So coming to the my instrument, like which is we developed one. So here if you see, if you add some dilution like the buffer, okay, suppose you have the blood sample and if dilute it, okay, accordingly, and you can use directly. So you no need of any reagent and straight away you can use for the measuring of absorption properties of those biological cells. So a small observation about like the quick observation about like the different literature reviews and what we need for the point of care device we can see here. So malaria, mostly malaria diagnosis is being performed by either the microscopy test, microscopy or RTD. Okay, and the flow cytometry and imaging flow cytometry has the capability, but not in use due to cost per test is high and they are bulky. So few of the point of care devices are developed and they are developing. Okay, and imaging flow cytometers are they are developed, but they have the limitation of the throughput. That is like the maximum you can have the 30 frames per second only, right? So this is the one. So from this, like what we got the conclusion is the simple technique to diagnose the malaria. Okay, and sample preparation should be very minimal, like just to dilute, and you can use for the high throughput analysis, and a portable and automated system. Okay, and the minimum skilled persons can operate and diagnose the disease. This is the some of the like the key points which we have considered into the point to in the process of development. So how it was realized is the like the work it is so I would be talking about that. So this is the initial work as I have earlier mentioned about this. Like if you are doing some spectroscopy analysis, you are getting average information of million or billion cells. Suppose you are having the couple of microliters, it reaches the billion cells, right? So, and in other hand, if you are able to measure a single cell measurement, the sensitivity and the like the accuracy of the like the diagnosis will be drastically increased. So the to develop this experimental setup we have designed is you can see here like the so these are the key points that like the to measure that points. Okay, this is the schematic diagram of the experiment setup. So you can see here this is like the vertical line is like the transmittance like the transmittance uh, custom built microscope. Okay, so basically if you like the as a if you are an instrument guy, so if you want to know each and every component here, like the what they are, so this is the LED source that is used for the imaging, and the condenser lens. This is the lens which is basically two lenses you are having here, so that you will be having some 4F system. Then you can eliminate some of the scattered information or the some of the artifacts, which is basically when you have any LED, so there would be some inside, like you will be finding some filament, right? In general bulb also we used to see filament. So while imaging the system, these things will come into the picture here, and your image will be disturbed. Okay, so to overcome this, like there is a color elimination system, which is also called as a 4F system. So we'll be having the aperture and the condenser lens about that. And this slide is focused on a microscopic slide. And the microscopic slide is prepared in a such a way that, okay, a single cells like the cells are like the clearly visible. They are well separated each other. So a thin layer was prepared on a microscopic slide. You can see how the cells are clearly like they are having the separation. Okay, and then micro objective. So basically it is collecting that in light information which is having the cells, and then this is a beam splitter. So basically beam splitter is used to divide your light beam into two parts. Suppose this beam splitter is 50 is to 50. So then your light is more or less it is dividing into two parts of the like the suppose here it is the 1 milliwatt. then 500 microwatts here and 500 microwatts here as it goes. And if your beam splitter is like the 70 is to 30, then accordingly it will be like the light ratio will be changing. Okay, and it is a tube lens, and the ND filter basically used for uh, like neutralizing the light. Suppose your camera is having capability of like the certain light range only. Okay, so if beyond that, then your light is, so you can see here some of the saturation which is reaching to 255 point. So when you have such kind of the information, then it is like the like you cannot extract the informations. Okay, so for that actually we used this neutral density filter. Okay, and the condenser. So basically, this is the microscope setup. Generally, when you have the custom bright field transmission microscope, this vertical line only you will be having. So what I did is like that I have slightly, but my thought was like that I want to develop a spectroscopy, right? Like the absorption measurement instrument. So absorption measurement instrument I supposed to develop. Okay, so in that case I have to uh, uh, sorry, I have to design this one only. I have to design this one only if I want to measure the absorption. But how do I know the single cell and its absorption I am able to measure? Okay, so for that I need some microscope also. So I have combined these two and made one system so that you can see the same cell, its morphological features here in the camera, and what is the change of the intensity taking place I can see in the photo detectors. That is the idea, like the how I developed this system. And of course initially when we are developing, there are like the different type of the problems arised in between, and we are unable to find the cell. And okay, so but like the later we have fixed those problems, and we successfully imaged single cells and their absorption properties, and we found, so you can see here that experimental setup which is we developed. So you can see here, this is the microscopic slide is present here on top of this microscopic slide, and this is the like basically XYZ translation stage, so that you can move your sample object like the microscopic slide from like the up, down, and like the side also. Okay, side as well. And this is micro objective, and as I mentioned that beam splitter, this is the beam splitter. Okay, tube lens which is we are used here, that is one, and the camera it is reaching here. So this one is the like the light intensity which is the laser source which is we are used, and this is the LED light which is used for the imaging purpose. So someone might be thinking like why you use the laser beam here only like the UV light which is close to 405 nanometer. Okay, and why use this? So basically, if I want to bring my sample into the focus, okay, when I want to bring my sample into the focus, then this is the the setup is for the single cell detection. Okay, single cell detection, right? So my beam spot supposed to be very narrow, which is around 10 to 12 micrometer. Okay, this laser beam, generally when you have any laser diode, if you see around 1 mm to 2 mm beam diameter, you can identify, right? You generally observe. So, but I made that 1 mm to 10 to 12 micrometer beam diameter. Okay, to focus on a small single cell, so that like the beam dias should not like the cover more than two cells or more than one cell, and it's supposed to cover one cell perfectly, so that I'll be getting that the single cell and its absorption property. Okay, so that was the the motto behind that, and why I choose this 405 nanometer laser is, so for example, if you see red blood cells, okay, RBC is generally this prone to, it has the hemoglobin content. Okay, hemoglobin content inside that. And suppose it is a reactive with like the, there is a highly absorbance at 420 nanometer, close to 420 nanometer, and around 530 nanometer or 540 nanometer range. It has the high absorptions. Okay, so uh, getting 420 nanometer for me, it was like the slightly tough, and it is available that laser pointer at 405 nanometer, and which has like the around 10 to 15 nanometer of like the plus or minus variation. So it served my purpose. It served my purpose, and we used that with this. So initially, what I have done is like we prepared a microscopic slide, and which has only normal human blood samples, which is having the only RBCs. There is no WBCs and any other things. Okay, so and like the initially we have visualized under this developer setup, and so basically when cell is present and when there is no cell is present, two areas I have identified, and properly where the cells is not present, that area I have identified, and like the single cell which is not covering with the multiple cells I have found, and then this laser source was illuminated on this area, and we found that okay, this is the intensity of the that particular uh, when we are having the cell, and this is the intensity we observed, and then we use this translation stage to move aside. Okay, translation stage to move aside, and we found that there is a huge difference where there is no cell is present. Then we found this much change of the intensity. So there is a like the huge change of the intensity when the cell is present and when there is no cell. So we have tried it multiple times to identify whether it is the like accurately giving the same information or not, and we found that okay, with the hundreds of cells we measured with this process, and we concluded that when cell is present and when there is no cell is present, there is a huge margin but absorption taking place. Okay, so with that we concluded, and then we have like the prepared another sample. Okay, we have prepared another sample which is having the malaria infected cells, and in combination of like the healthy cells and the malaria infected cells, both cells are present. Okay, both cells are present, and so we found the locations accordingly, that where suppose for example I have laser source and here one cell and this is one. So this is the malaria infected and this is the normal. So suppose initially it is at center, and then I have focused this light on this. So then we've observed that this is the change of intensity took place, and this is the healthy cell when like the mood towards this in this direction, and the cell is in the focus, then we found that the change of intensity is this. Okay, so you can see for like the reconfirming and rechecking purpose we have done it multiple times, and it is evident that from various other studies also. So when RBCs, red blood cells, are affected with like the malaria, so malaria parasite which is present inside that, okay, it is consuming this hemoglobin content, okay, which is present in the hemoglobin in a RBC. This parasite is consuming and surviving inside that, and it is trying to multiply inside the RBC. Okay, so and normal healthy RBCs which is having the hemoglobin content, and this light source which is like the light sensitive with this particular wavelength of the light. Okay, so it is obvious, right? We can find that the change of the intensities when the cell is present and when there is no cell is present, when cell is present and infected cell is also present. So this gave us like the boost that okay, we can develop one based on the absorption properties, we can use like the develop instrument, okay, which can be used for the malaria detection. But as I initially mentioned that this is the throughput limitation is there. So to overcome that throughput limitation, you can see here that from smear to inflow with the help of the microfluidic platform. So it is like the crucial step to make it the high throughput analysis. So the design and development of the microfluidic device with having the channels dimension of like something around 8 micrometer wide and 5 micrometer deep. Okay, 5 micrometer deep. This is the channel dimension. So you can see here the single cell how it is moving. And accordingly, the change of intensity I have measured with this. Okay, so design and development of the microfluidic devices, we can see like it has like the huge applications. Okay, like the And the advantages, small length in scale, like that you can use the very less reagents, and the sample quantity also in a microliter of the blood sample you can sufficient for identifying like the disease, any type of the disease and detections. Okay, and different type of the flow and like the flow focus analysis and so on, various applications we have here in the microfluidic devices. So this is also like the shows that like the we can use in the various applications, like either it is the diagnostic, molecular and cell passivling DNA, RNA, protein finding purpose, and many more applications we can find. Okay, so this is like the there is a kind of the information about the microfluidic device, how can we use. So uh, so basically here like for single cell analysis we need, okay, single cell analysis we need a specific device, okay, specific device where we can flow the single cells. For example, if you see here, it is something around 500 micrometers or 200 micrometers, it is the size. So you can see here the multiple cells, but that is not serving my purpose. So I made a channel in a such a way that, and which is going down to the narrow down, where like we can have around 10 micrometers or 12 micrometers micro channels, okay, 10 micrometer wide micro channels, and so that like the my intensity of the light which can sufficiently cover, so the no cells can escape from this intensity, and this is the outlet. So this is the design of the microfluidic device. And okay, let me check what is the time we have. Okay, it's almost done. So maybe I'll quickly wrap up it. So if you see the design and development of the microfluidic device, it involves various steps, like the design. Initially, you need to design the mask and design through AutoCAD, right? AutoCAD, and then once the mask is prepared, then you have to use that mask to fabricate master. Master is like it would be on a silicon wafer, and then it's supposed to be reflected with the, like the PDMS polymer, so that like in the final step we will be getting like the microfluidic device is like this. So initially you will be getting from here this kind of the microfluidic device, and when you bond with then it is having like the three side it is open, three side it is closed, and one side it is like the open, right? One side it is open. So one side open area, after inserting the inlet and outlet, you have to bond this with the cover slip, so that like the microchannels will be covered fully, right? Then the sample can flow through this end, and like the sample will come out from this outlet, okay, generally. So this is the process is involved. So you can see here the complete photolithography process, how generally the development of the microfluidic device takes place. So initially photolithography, which is developing you a master on a silicon wafer. Okay, then you have to follow the soft lithography technique procedure, that is the PDMS, and then mix it. Then you will be finding some bubbles inside that. And when you use the vacuum desiccator, okay, when you use vacuum desiccators and like the vacuum pressure, when you use, those bubbles will come out, and they will get busted, they will go out. So once they are bubble free, when you have this, then you can pour on this master, which is you are getting here after the development. Okay, then to solidify it, like you have to bake it and then peel it out. Once you peel it out, then you will be getting the three sided, it is closed, but one side it is open, which is also we can call it as a mold. Okay, and then when you bond with the cover slip, then your microfluidic device is ready. So, like if you see the initial schematic diagram of the experimental setup, and here there is a slight change. So initially I was using the microscopic slide, right? Microscopic slide, and here it is a microfluidic device. So in a microscopic slide, you have to inspect like the various location of the microscopic slide for identifying the each and every cells. Okay, but in this case, microfluidic device has the micro channels. Okay, once you are connected with the like the some inlet and outlet, so sample will be flowing through it. So once you have like the arranged it aligned with the, like the light which is you are focusing, so then no need of to move, no need of any movement in the microfluidic device. So that is the reason the single source I have used for this. And you can see like it is a kind of the like the some of the arrangement. So you can say it is a 4F system, so that the collimated beam you will get, and then again it is by using another lens you have focused, we have focused it into the spot, tiny spot, and then the micro objective, and appropriately we were having beam splitter like here. For the one is the detector and another is the camera. So like this was like the realized as a hyper imaging system for the calibration. Okay, and various other application also, deformability and other thing. So why we are calling here is hyper imaging system is, suppose any system if you consider microscopy means you will be getting the morphological features only, this one only, right? And if any spectroscopy analysis, you will be getting only the change of the spectrum only, right? But this system, you can get both the information and in a synchronous manner. So both the information you can get in the synchronous manner means like one cell is passing through it, and this what is the cell, and like the what is the morphology of that cell has, and like the what is the change of the intensity taking place. That information also you can get. So this was used for like the high throughput characterization purpose. So you can see here how we have characterized. Okay, so you can find here the cells is moving, and those are the single cells at a time. Okay, cells, and accordingly this when the cell come here, you can see here one cursor, right? So at this point you can see the change of the intensity. Okay, so when the cell is passing, and accordingly you can see the change of the intensity. So like we have identified each cell and its change of intensity. What is the change of the percentage of the absorption taking place? We have identified, and based on that, we made a threshold for like the healthy and like the malaria infected cells, their absorption properties. So we found that the intensity of the absorption around 4.5 was there with the, like the when you have the infected malaria infected RBCs and healthy RBCs, like it was close to 10% of the absorption taking place. Okay, so this was gave the like the us like the clear information that we can find that the change of the intensity at a high throughput manner. And you can see here like the change of intensity with two different samples. So basically here the red one is like the having the both malaria infected RBCs plus healthy, it means normal RBCs, and this is the one like the area where we have like the complete healthy RBCs, normal RBCs. So when we overlapped these two data, so then we found that the clear information with this. So zoomed area you can see here. So this is the area where like the change of the intensity is less, and it is matched with whatever we got the clinical microscopes data. Suppose for example we have got 1% of the blood cells was like the malaria infected. It means out of 100, one RBC is infected. Uh, without like the whatever the data we found here, which is quantitatively, that is also matching. So it gave us like clear information that we can identify the malaria infected and healthy RBCs here. And this is are like some of the like the different cases where we found like some difficulties. Okay, so why we need like the hyper imaging system for the better characterization and other things. Okay, so this is the one case, right? So where we can see the two different peaks, down peaks, but here we don't see like the like that, but it is a kind of the both the intensity is combined, and this is the case where we have the single RBC. So this hyper imaging system gave us the clear information that what is the like some two or more cells are overlapping, what would be the intensity. Okay, and when we are having the single RBCs, then what is the intensity. Those all information we found, and that was the one of the advantage with the hyper imaging system. Of course, with this we can identify the threshold of like the which is the different cases, right? And we can reject those signals specific to the like the applications orientation. So we have assessed about like 300,000 cells within the 3 minutes of the time duration. And you can see here that is the blood samples where like the we were having it is equally matching with like whatever we found with our like the absorption analyzer histograms and the clinical microscopy data. So and we found that okay, this was like the slightly the sample was having the higher like the next stage, it is not early stage. So with respect to that, suppose when you have the early stage, then we used to get like the this place like the absorption changes place. But when we are having the late stage some information, then we found that okay, the absorption is very poor, like very poor in that this case. So they are those cells actually they are multiplied. The parasite is high in the level of in those areas. Okay, and the final step with this, actually, we have found that the threshold for malaria infected and the normal RBCs. Okay, and by using that, actually, what we need to realize that develop an instrument that is source sample detector was we moved towards like the casing of those instrument. Okay, so and all the essential components which is we used for the like the experiment, like laser source and like the condenser lens, microfluidic device, micro objective. Okay, micro objective, then the beam splitter. So basically, this beam splitter and this camera is not required as per my statistics, but so we should it should work with this only. And in real scenario, it's supposed to be work like that only. But why we have used this like a simple camera Raspberry Pi camera is, suppose I have micro channel is 10 micrometer, right? This is the micro channel which is having the 10 micrometer diameter. So with the naked eye, if you want to align that micro channel and the laser beam, it is very tough. Okay, it is not easy, and you are not sure about that. So to overcome that, suppose a initial simple camera will help us to the micro channel and this laser point spot is in alignment or not. They are in the like the proper place or not. You will see, and then the rest of the whatever the application which is we have to diagnose and identify that data collection and analysis will be carried out with the detector and like the simple microcontroller which is the Raspberry Pi. Mario we use right yes here. So this is the like the some simple like the flow chart of the like the how we have collected the data you know, from like the, using this detector and microcontroller. So initialization and the SD card we have to add and the initialization. Once the initialization done, then it's supposed to create a file. Okay, then it collect the samples, and the, once the sample collection is done, then it move to the next step. So this process will continue. Okay, so this is like due to the time constraints, I'm like not in details I'm going here. Okay, so you can see here like the few more like the analysis which is we have found like the data we acquired by using our system. Okay, and we can find here like the malaria infected cell and its absorption and healthy RBCs, and that this is the threshold for that. Okay, so as of now, I was talking about like the malaria infected and healthy RBCs as of right, but we know that like the blood sample contains everything like WBCs, platelets, right? RBCs, WBCs, and platelets, everything. So what about the what is the absorption properties? It is coming into the picture in case of the WBCs, and what is the absorption properties coming to the picture when you have the platelets? Okay, and of course, we have talked about the RBCs and healthy and normal malaria infected RBCs and what is the absorption takes place. That also we have discussed, but these two not right. So to identify this, actually, I have run a couple of tests related to that, like when I'm having the platelets and WBCs in the sample, and I have run the test and found that like we don't find much change of the intensity when WBCs and platelets were present. Same time, like the only the I have collected the platelets like the from the upper layer, so where we have the mostly platelets only, and there also we found the similar things. Okay, and Then we have also tried that, like suppose only buffer you are flowing through the micro channels, what will be the change of the intensity taking place? Is there any noise present? Okay, for that we have like the conducted, and we found the as not so many these peaks, but less similar to that. And only PDMS is present. There is no sample is flowing. There is no buffer flowing. Then also we found some couple of things like this. So what we conclude is here, this is less than it. This is the threshold of less than 1%, okay, of absorption. So maybe this is due to the WBC or platelets or just noise of the source, laser source. Okay, so we don't know. So that is the reason actually we have prepared one upper threshold so that this kind of the this kind of the like the unwanted data we can eliminate. So this is the upper threshold, and then the final experiment we have like the final kind of the final experiment we have done, where we have like upper threshold, right? This is the noise level and upper threshold, and this is the lower threshold where we have this is for the malaria infected RBCs and healthy RBCs. Like below is the healthy RBCs, right? Below is the healthy RBCs, and this is the area where the malaria infected like the region. Okay, and this is the level of the signal where we can get the like the peak. Okay, so it gave like us like the prominent results where we compared to like the what we found with the clinical microscopy, same data it gave, more or less more. And so advantage with this system is, suppose like the 100 infected cells are present in the like the sample, it shows that 100 are like the having like the change of intensity is this much. Okay, and like the remaining all are the change of the intensity is this much. Like you can find this. But coming to the like the suppose you have like the microscopic clinical microscopy, what we assume there is, okay, in this field of view I haven't find, in this field of view I have find one, in this field of view I have find like the two infected cells. So overall maybe it is a 1% of like the malaria infected cellular cells were present. Like that you can conclude. Okay, so but here you can get the quantitative information, how many cells are present when, and how many cells, like, suppose you have like the total cells you found 10,000 cells, right? Out of which you are having the 100 is infected, then you will find 100 infected cells in this, right? Okay, so to summarize that work, like the demonstrated a technique to characterize the optical absorption of cells on a single cell basis from the given population. And it was like we are able to measure that like the like optical absorption of malaria infected and healthy RBCs from the given cells. Of course, what is the limitation we have here? We don't find the WBCs and platelets. What is the like the PLTs, platelets and WBCs, like the what is the quantity they has, that we couldn't find. That is the limitation. But the RBCs and malaria infected RBCs were clearly identified with this, and this system is completely label free. Just you can use, like suppose you collected 1 microliter of the blood sample and you added like the something around 20 microliter of the buffer. Okay, so you have diluted it and used for the testing, and it is done. So there is no need of any reagent, either it is a methylene blue or it is the fluorescence dye or anything. And like the finally, actually, we have made a prototype which can be used for the clinical, go for the clinical evaluation. But of course, there is like the there are TRL, whatever my professor previously he told, like different stages of the things are there. So as of now, we have prepared that, and it was stopped there the work. And then I came here, and so now I'm going to pursue various applications by using the same system. Okay, same hyperimaging system and like the point of care device for the malaria detection. So ultimate goal of this instrument, it's supposed to be like any person is having fever and reach to the pathologist. Okay, when he reaches to the pathologist should be in a position to tell, okay, this is just a viral fever, this is the dengue, or this is a malaria infection, and give accordingly the prescription. Okay, that is the idea of this like the work which is we are like the pursuing. Okay, so I hope that will be uh, completed in couple of like the soon. That's what we can say. And there are different other applications also we can use. So for example, deformability measurements. Okay, so hyperimaging system where we have like the you can see the imaging also and the single cell analysis, right? A single cell, okay, flowing through the microchannel and with a flow rate, desired flow rate. So what is the stretch is taking due to the pressure of the flow, right? That can be like the carried out by using this system, a hyperimaging system, not absorption flow cytometer, which is like the which is the system we use for the characterization. And some of the more other applications related to the microfluidic aspects and other things. Okay, as we know, suppose if you want to go for the drug screening and delivery applications, we used to use like the animals, right, for that, to identify different determine the toxicity and the efficacy of the animals and various things. Okay, so initially we used to like the use for rats and different other animals, and then we identify a couple of related like the data or the statistics. So instead of that, you can like the do the similar applications. For example, you have cells, blood cells, and you have suspended on a microfluidic device. Okay, and you entered into the drugs into that, right? You have entered into the drug, and you have samples here. So this drugs is when it is passing through it, and you have the laser source and detector arms. Okay, so what is the morphology is taking place when you are adding the drug, and what is the like the it is in the like whether it is the cells is still surviving or the multiplying inside that or not. So those things, this kind of the application you can use. So what is the advantage compared to this? So like we know that when you are like the maintaining this kind of the like the facilities, we need to have a lot of maintenance and other things, and we need to many more important is like we have to sacrifice this right, for the various application. But coming to the here, just if you have the sample, then you can test on a microfluidic platform, so that it is like the more easier, and it is easy to do the different type of the analysis, right? So with this I thank you, my work is end here. So these are the a couple of applications which we are going to pursue here in the like the NIT Raurkela. And so I'm happy to show here like the my lab, and thank you. Like if you have any questions, feel free to ask. I'm happy to answer you.
Thank you very much, sir, for sharing your experience. It was very insightful talk. If there is any question, floor is open. Okay, so if anyone is having any question, they can even
Just
drop in the chat
Sika, I
box,
can
or I can
ask
let.
one question.
Okay.
Maybe, sir, you can explain like maybe like when we are doing, in the flow channel, blood
Okay.
RBCs, WBCs, all sorts of cells are passing. So how do you identify RBC and WBC or distinguish between them?
Okay. So like you saw, right, like one thing like the micro channels when you are having, and the
Yes.
change of the intensity also you are seeing, right? When you
Okay.
have when you are collected data, right? So you have collected data, and frame by frame when you see that image processing or whatever you do, suppose you found that the major axis and minor axis, and you know like the what is the size of the RBC, what is the size of the WBC, right? So for example, WBCs are like their size is close to 10 to 15 micrometers, and the RBC size is like 6 to 8 micrometers, right? So based on that, like when you find that major and minor axis, and accordingly, if it is matching with the the size of the whatever you know, then you can say that this is the RBC and this is WBC. And one more thing is like if I haven't mentioned here, but when you use this particular wavelength of the light, okay, you can clearly identify that the hemoglobin, like the darker part. Okay,
Okay.
I used to so when you are seeing some cells are flowing in a disc shape, you used to see that something like kind of the disc, and those are like the very concentrated. And but coming to the WBCs, it would be more transparent. With respect to the light also, different wavelength of the light when you are using, okay, that also gives you like the morphological informations clearly.
Okay. Okay, sir. So I like it's a combination of morphological features and some properties like whether the intensity is changing or not, and these kind of
Yes, yes.
things. Okay, great. Okay, thank you, sir. The floor is still open for questions. If any questions, we can take. Okay.
Okay.
So thank you, sir, once
Thank
again for.
thank you,
Okay,
sir,
sir.
and thank
Yeah.
you, Kali sir, like
Thank you very much, sir.
for
It was a
Thank you,
hosting
sir.
wonderful
me.
session.
Uh,